let's move on to our next topic, that is microscopy. And you all know microscopy is the technique of using microscope to observe the structures which are not visible under naked eyes or unaided eyes. So here we are going to discuss mainly three microscopic techniques, which are optical microscopy, electron microscopy, and X-ray microscopy. So let's discuss each one by one in detail. So we have our first microscopy, that is optical microscopy, and this microscopy is also known as light microscopy because we are using visible light here to create the image of an object. And this visible light may be either from a natural source or it may be from an artificial source. Okay. And here we can use simple or compound microscope. So any of these two microscope can be used to perform this light microscopy. So what is the basic difference between these two microscopes? So in this image, you can clearly appreciate the difference. So this is simple microscope and this is compound microscope. And the basic difference is that simple microscope is made up of single lens. Here you can see the single lens. Okay. While the compound microscope is made up of two or more lenses. Here you can see objective lenses of different power and eyepiece. Okay, so this is the basic difference between a simple and compound microscope that can be used to perform optical microscopy. This optical microscopy is the most common type of microscopy among all techniques. Okay, among all microscopy, this is the most common type of microscopy we are using regularly. Okay, we are using performing optical microscopy in our routine practical examinations. Okay, and we can produce 2D images only by this microscopy. We cannot produce 3D image. Okay. While discussing the microscope, the two definitions are important, and these two definitions are magnification power and resolution power. So what is magnification power of a microscope? So as name indicates, magnification power. It is ability to magnify. It is the ability to enlarge the image of an object is called magnification power of a microscope. And resolution power, it is ability to resolve. It is ability to differentiate between 2 points that are separated at a minimum distance is called resolution power of a microscope. Okay. So if we talk about the magnification power of simple light microscope, it is 10 to 20 times. Means the simple light microscope can enlarge the image of an object up to 10 to 20 times only, not more than this. Okay. While the compound light microscope has a magnification power up to 1000 times, means it can enlarge the image of an object up to 1000 times, right? If we talk about the resolution power, so the resolution power of simple light microscope is 1 micrometer, means it can separate the 2 points. It can differentiate between 2 points that are separated at a minimum distance of 1 micrometer. Okay. While the compound microscope has a resolution power of 0.2 micrometer, means this compound light microscope can separate the 2 points that are separated at the distance of minimum 0.2 micrometer. Okay. So suppose we have two bacteria. First is this and second is this. Okay. And these two bacteria are separated from each other at 0.5 micrometer. And if we want to see these two bacteria separately, so what do we need? Do we need a simple light microscope? No, because the resolution power of simple light microscope is 1 micrometer. And here the distance between these two organisms is 0.5 microscope. So if we are using light microscope, we cannot see them separately. These two will be visible as a single under simple light microscope. Okay. So we need to use a compound light microscope to see these two bacteria separately, because compound light microscope has a resolution power of 0.2 micrometer, right? Now this optical microscopy is of 4 types. First is bright field microscopy. Second is dark field microscopy. Third is phase contrast microscopy. And fourth is fluorescent microscopy. So let's discuss each and every technique one by one. Okay. So first optical microscopy is bright field microscopy. See, field means background, and here the background will be bright. Okay. So in the bright field microscopy, the background will be bright. Okay, and here we can use a simple or compound light microscope to perform this microscopy. Clear? This is the simplest and most common type of microscopy among all optical microscopy. Okay, so don't get confused. Optical microscopy is the most common type of microscopy among all microscopy, while the bright field microscopy is the simplest and most common microscopic technique among all optical microscopy techniques. Okay, and we need to stain the objects we are going to observe under this microscopy. Okay, so we need staining technique here. Okay, so staining technique is required in this microscopy. Next optical microscopy is dark field microscopy. As name indicates, the field will be dark or background will be black. Okay, and here we are using a special compound microscope which is equipped with a special dark field condenser which is fitted with an opaque disc or central stalk. This is a special type of compound microscope, and it is fitted with this opaque disc or central stalk. This central stalk will block the direct light, and once the direct light is blocked, the background will be black. Okay, so here we will get a black background, right? So whatever the scattered light, the light will reach to the objective lens and will lead to the formation of image of that object. So this is the functioning of dark field microscopy. We do not need any kind of staining here. Okay, so we do not need to stain the objects. We can go directly. Okay. Now this this dark field microscopy is used for motility demonstration. So if you want to demonstrate the motility of a bacteria, you have to go for dark field microscopy. Very important point. Which microscopic technique is used for motility demonstration? The answer is dark field microscopy. Second use of this microscopy is the demonstration of spirochetes. Whatever spirochetes we have Treponema, we have Leptospira, we have Borrelia. If you want to demonstrate these organisms, you need to go for dark field microscopy. These are easily demonstrated under dark field microscopy. Okay, so please remember these 2 points. For the motility demonstration, we can use dark field microscopy. For the spirochete demonstration, we can go for dark field microscopy. Third optical microscopy is phase contrast microscopy, right? And here we are using a special kind of compound microscope, that is phase contrast microscope. And this microscopy is based on the principle of refractive index. So what is a refractive index? Refractive index is the property of an object to bend the light when light passes through it. Okay. So we have a cell, and cell have different components, and different components of cell have different refractive index. Okay. For an example, if we are talking about cytoplasm, so cytoplasm has low refractive index and it will appear lighter under phase contrast microscopy. Clear? Similarly, nucleus has a higher refractive index and it will appear darker under phase contrast microscopy. Clear? We do not need any kind of staining here also. So if you want to observe live cells, if you want to observe the internal structures of a cell, you can go for phase contrast microscopy, because different components of cells have different refractive index and we can easily differentiate these internal structures of the cells by performing phase contrast microscopy. Okay. The last optical microscopy is fluorescent microscopy, and here we are using fluorescent microscope, which is a special type of compound microscope. And this microscopic technique is based on the property of fluorescence. Fluorescence is the property to emit the light. Okay, so here we are going to stain the objects with fluorescent dye, and once the object is stained with fluorescent dye, it will start emitting light. And this emission of light we can easily observe under fluorescent microscope. So this is the fluorescent microscopy, and this microscopic technique is used mainly for the detection of antigen and antibody in immunology, and we can also use this technique to visualize the DNA, RNA, and proteins in a cell or tissues. Okay, so please remember the use of this microscopic techniques, right? Next microscopy is electron microscopy, and here we are using beams of electrons instead of light. In the optical microscopy, we are using visible lights to create the image of an object. But here we are using beams of electrons to create the image of an object, right? And here we are using electron microscope, and we have two types of electron microscope. First is SEM, that is a scanning electron microscope. Second is TAM, that is transmission electron microscope. So this scanning electron microscope will scan the surface details of the object, and it will provide only surface details. Okay. While the transmission electron microscope will transmit the electrons inside the objects, and it will provide internal detail. So this is the basic difference between two electron microscope, that is a scanning electron microscope and transmission electron microscope. Okay. SEM will provide only surface detail, while TEM will provide internal details. Okay. Now, if we talk about the magnification power and resolution power, so the magnification power for the SEM is 50 million times. Please remember this. And the resolution power for SEM is around 1 to 10 nanometers, right? For the transmission electron microscope, the magnification power will be up to 1 to 2 million times, while the resolution power will be around 0.2 nanometers. Clear? And the use of this microscopy is to analyze the structures of bacteria, the structure of viruses, and the detailed structures of cell. Okay. So if you want to analyze the structure of bacteria, if you want to go for the study of viruses, you can go for the electron microscopy. And we can produce 3D images of an object. Okay, we can produce 3D images of the objects by this microscopic technique. Okay, so this is about electron microscopy. Now the last microscopy we have X-ray microscopy, and here we are using soft X-ray radiations. We are not using light, we are not using electron here. We are just using soft X-ray radiations here to perform this microscopy. Clear? Now, if we talk about the resolution power of this microscopy or this X-ray microscope, the resolution power will be greater than light microscopy, while it will be lesser than electron microscopy. Clear? This microscopic technique is not a common approach. So we will not go in detail of this technique. Okay. So this is about the microscopy.